All right, we are now recording, and thank you very much, Tobias, for agreeing to give a Build-A-Cell seminar, and you can get started.
Yeah, so thank you for inviting me, first of all, to this seminar series. I hope that you all get something with you from my talk. So if there are any questions, I think we will have a question session at the end of the talk if there's something really urgent. So don't hesitate to interrupt me before the rest starts getting unclear. So I was asked to introduce myself at the beginning, which is always kind of difficult. So unfortunately, I have no cat, no dogs, as we already discussed. So I'm just sticking with the facts. So my name is Tobias Neckernuss. I did my PhD in physics, and also I studied physics at Ulm University. So it's a small town, more or less in the south of Germany. And right now I'm the project leader of the Exist-Forschungstransfer at the university. And at the same time, I'm the founder and managing director of the Sensific company, which was founded for getting the results that we did in research over the last years into market. I will come to this point a little bit later in the talk. So the most important partner or most important people in this talk who contributed the most are all located at Ulm University at the Institute of Experimental Physics. So it's the head of our department, Professor Ottmar Marti, and the two PhD students, Patricia Schwilling and Lisa Quappich, and my two colleagues, Jonas Pfeil and Daniel Geiger, who also founded with me this company. And these people here that you see here did most of the work. So especially most of the lab work that you are going to see was done by Patricia Schwilling. Most of the programming was done by Jonas, and most of the hardware and electrical designing things were done by Daniel Geiger. And as I already introduced at the beginning, we founded this company, Sensific, with the aim to bring this product and the techniques that we introduce, or that I will introduce right now, to bring them into market. And the product of this company is this Odin device that you already saw on the first slide, but I will come to this all a little bit later. So let's start with a big thank you at the beginning, so everybody is still listening. This is very important because I want to be really thankful to especially the Institute of Experimental Physics with Professor Ottmar Marti, who always let us do what we thought is reasonable and what we thought is interesting. And we are very grateful for his support and being in the institute with him, together, of course, with the two persons I already introduced, our PhD students, and the rest of the staff of the institute. And of course, to our, let's call it major cooperation partner from Max Planck Institute for Medical Research in Heidelberg, Professor Joachim Spatz, Ilja Platzmann, Christoph Frey, and Manuel Baumann, who also contributed to kinds of this work. And of course, everybody needs money, so I'm really thankful to this great funding partners of the work, and who are taking care that we also can buy something if we need. Okay, so I think that the people here are from a broad background, but I think I don't have to introduce the thematics of droplet-based microfluidic. I think over the last 15 years, maybe, this topic evolved so much and is still so much evolving that every day you can read tons of new articles if you like. So the general idea is that you can encapsulate things, objects, cells, bacteria, proteins, DNA, whatever you like, in small compartments and handle them in a fluidic way without getting intermixed with each other, and having complex lab-on-a-chip arrays of materials that you can work with. So for all these applications, I want to quickly go through a few of them because I think from my opinion, it shows perfectly the wide variety of microfluidics and droplet-based microfluidics, what you can achieve with it. I think digital droplet PCR, I don't have to explain right now. I mean, PCR is in everyday's mouth today due to the COVID crisis. So there are different things. So normally you do bulk PCR things. That's what is normally done. However, you can also do this, for example, in very, very small compartments. So you compartmentalize your sample together with your reagent in small liquid droplets in a liquid fluid phase. You can do all the cycling and after all the readout, and then in a special thing, what you can only do here and not so much in the bulk thing is that you can have a lot of samples cycled and analyzed over each other in a fluid way. So you have a really, really high throughput. Then one thing that's also very well evolving over the last years is the directed evolution of screening. So if you want to manipulate for example, let's take the bacteria, you want to have a new bacteria that is capable of doing something very well. So you start with an old bacteria and you do some genetic engineering, or you just let it do by them own. And then you compartmentalize it, put it into small droplets, and you see this bacteria or your sample is doing well or not so well. So you can sort out all those bacteria that are doing well and get them feedback into the first process, repeat this process so you get faster to your results. So other techniques that are very, very prominent in droplet-based microfluidics are, for example, targeted cell binding antibodies, so that you can directly say which kind of antibodies are in your sample, which kind of antibodies bind to specific cells or to specific binding sites, together with a very, very new technique. So I think it was published 2 years ago, so which is a digital droplet ELISA, which is also very promising because you can get faster to your results and need less amount of sample and can later on handle your sample very, very well. Of course, then not only on a molecular level, but also on a cellular level, what it can be done, for example, is T-cell therapy research, where you encapsulate cancer cells together with target cells and see how they kill each other and how well this process is happening. And then you can learn something for cancer therapy and all the stuff. So I think for all these techniques that are very well known and applied by hundreds or even thousands of groups worldwide, you have some needs and some demands that each setup where you handle these samples and where you're doing these techniques need to meet. So the first thing you really have to worry about if you're doing something like droplet, let's call it all over it, droplet microfluidics, is you have to investigate your system. And there are some prerequisites for doing this properly. So first of all, the good thing on droplet-based microfluidic is that you have a very high throughput. So also your screening or imaging or detection method has to be with a very high throughput. Then you want to be, of course, single cell or at least single droplet sensitive, because you want to analyze each and every droplet that you have in your system. If you're not 100% sure what you're doing, which is quite often in research, then you're always grateful if you also have an optical feedback of what you are doing. Are the droplets looking well? Do the cells look okay? Is the color of the fluorescent okay that is inside of the sample and all the stuff? So you want to have something that you can also see by eye. Then there are techniques where you need to label your cells because you want to be really binding specific, or you want to be specific to a specific chemistry, or you don't want to label them because you want to later use your results of your cells, for example, and work with them further, and then labeling would be of disadvantage. So your detection system should be able to do both label and label-free detection. Of course, it's always good if your system is easy to implement, everybody can use it. Then there are, in principle, two types of setups. So one is an add-on setup, which you can use like a camera, for example, for your existing like Microscope, where you just can put it in. The other thing is a totally standalone setup, which is more or less boxed in and everything happens automatically. So you don't have to worry about, but you also cannot interfere with the measurement if you like, or you cannot so much work on the measurement process itself. And of course, at the end of the day, we all have to be cost efficient. So you should get a decent value out of the instrument for the price that you pay for it. So in the following slides, I will have a quick review on what is on the market or available for detecting, imaging and sorting in microfluidic systems, and what are the advantages and also the disadvantages, or just the specificities of those instruments. So first of all, classical thing is you collect your sample, you collect your droplet, put it into something like a well plate or a microfluidic well plate in this case here, and you have a look over your normal microscope. You can use a normal microscope camera, and you can tell, okay, here is a very small cell, there are 4 cells encapsulated, and so on. And then if you're lucky, you have a gating system where you can put through those droplets one by one and decide what's happening with each droplet. Or if you're not so happy, you have to use micropipette and pipette it out of these wells manually. So of course, you have a visual feedback, but it's very, very slow. You have a human bias by calculating or by judging the quality of your sample. And it's very, very slow, and you have a very low throughput. Second thing, which is more elaborated, however, still very easy to implement, is impedance measurements. So you're measuring the impedance or the change of impedance when a sample or a droplet runs through an electrode system like you see here. And from the specific change of the impedance, you can already calculate back or guess back on specific properties of your sample. So this is, for example, done if you go to a medical doctor and have a small blood count, so just distinguishing between white and red blood cells, for example. This can be very easily done with this setup. However, you got no optical feedback, and your the amount of information you get from your sample is quite limited. Then, of course, what is often used in microfluidic systems is high-speed video microscopy. The good thing is you see almost everything that you can see if you have a good contrast and if you have a good microscope. You have a large region of interest that you can observe. However, you cannot judge by eye or directly by the instrument. So you have to record a high-speed video, save it on a hard drive, and do the evaluation later on. However, there are good tools available, free tools for evaluating such videos, so you get very precise results. However, you have a time lag between recording or between the happening of the event and the result that you have. Then I mean, it's getting more and more more technically and more expensive also. You have a setup of flow cytometry. Here you have your sample in a capillary like here, so may it be cells or may it be droplets or other kind of particles. You shine a laser on it and you collect the forward and the sideward scattering. And then from there you get information about the scattering behavior of your sample. And with this, you can classify the object that you're investigating. The good thing here is that you can introduce fluorescent labels and be chemically specific. However, you will get no real, so in the normal instruments, you will get no real image of your sample. So you don't know how it looks like. And the information of the bright field information, so the non-colored or non-labeled information is not that much. So one step further is the FACS, the fluorescent activated cell sorting. Here, the principle is the same, and you're also having an electrode system here, which you can gate. So you can say, okay, if my 2 detectors detect a specific pattern in my sample, I want to sort out all those, for example, that are shining bright green. and not those that are shining bright blue like you see here. So, and then you can sort out the green and the blue one according to your system. So normally those systems are encapsulated in a benchtop system or even larger. So you have to get in your sample and get out and be happy with what you have. So you have no real chance of interfering with the system. And of course, there are plenty of very specialized instruments on the market that you can buy and that you can use for a very specific purpose. They are very useful for your specific problem. However, they are only useful for your specific problem and not so much generalized for a general approach. So this is approximately what is more or less available. So might be self-built or whether you can buy it. And we thought that there's still a lot of space around. So and we were not happy with the existing solutions for the microfluidic imaging, detection, and analysis, because we wanted to have something that is able of investigating visually, so optically, what is happening on your sample, and on the same time being able to directly interfere with your sample. So if you have a specific event, you really want to make something happen in your sample. So might it be to inject something into the droplet, may it be sorting it out, may it be accelerating the flow or whatever. So therefore you have specific requirements. And this is what we developed over the last, so maybe now 4 years approximately. And the end point of this is the so-called Odin device. So in principle, it's quite easy. It's a high performance scientific machine vision system. So it's a camera which is capable of doing really high speed, however, still real-time detection. And you can analyze a lot of objects which we will see later. So how does it look like? So in principle, so this is our microscope here. And this is the Odin device. As you see, it's just mounted on the camera port and is approximately the size of a normal microscope camera, or as like a cup of like a coffee cup, something like this. This is the approximate size. So what is this exactly? So as I said before, in principle, it's just a fast camera. However, the data analysis is done directly on the chip of the camera. So we don't have additional hardware or additional software. So there's no additional computer where you transfer your data to. No, the data is directly analyzed and processed on the system. And this enables us to meet these real hard timing requirements that are necessary in order to interfere directly with your sample. You can mount it on a microscope, like on a C-mount, and it works completely autonomously. So as I said before, no additional hardware needed. And it is suitable for if you just want to have an image of your experiment, or you want a thoroughly documentation of your experiment, or where you want to steer and control your experiment. You can all do this with this new with this device. So so this is a sketch of a possible experiment that you can do. So you have your microfluidic chip on your microscope, like here. You can control the flow of the objects or of the different phases with a normal microfluidic flow controller, and you image your sample through the conventional microscope port, through the conventional camera port of your microscope. And then Odin does all the calculation and gives you the results of your measurements. And at the same time, you can tell the system or the periphery system, for example, here shown as a sorting system, what it should do. So if you have a specific event that you are interested in, the sensor can tell, for example, the function generator to start generating a sinus function, and which is amplified and brought to these two electrodes so that you can introduce the electrophoretic sorting. We will see this later. So the quick benchmarks are that you can see samples with 10,000 Hertz, and which then leads to a detection rate of approximately 3,000 particles per second. So over this sheet, I want to go briefly. So what makes it, so what is the difference between the conventional high-speed camera and the newly developed method is with a conventional high-speed camera, in principle, you start here, you capture your image, and you have an image stream or a data stream of approximately 300 megabytes per second. So, and at this point, it is already clear that that working with this data stream somewhere else than directly on the camera is almost impossible, because if you have to transfer these amounts of data and organize them on a external computer, then all your timing requirements will be lost. This is why we directly reduce the amount of data on the chip. So if you just think of a microscopic image, so most of the image information is not important for you. I mean, there's a lot of things in the background that's happening that you are not interested in. Then almost each and every image of your sample looks alike. So this can be also kind of compressed. And this is what is done on these on this system. It's all done on a FPGA, which is an electronic device for, so in principle, it's a microchip device which can do specific calculations and specific routings very, very fast. And after you did all these, kind of algorithms that are all developed by Jonas, you end up with some parameters classifying your sample as good as possible. So first we started with very, very basic parameters like droplet size, droplet shape, droplet velocity, and then after over time we evolved with these parameters and added some more, like different markers of morphology, for example, and also some empirical parameters that turned out to be useful in order to detect two different kind of samples that we came over during the evaluation process. And at the end of this system, you can either store your data like you did it before, or you can use this as a real-time trigger system in order to trigger some events directly happening on the lab on a chip system. So this was hopefully enough of theory. So now we see some measurement results. So this is just a benchmarking system. So these are just cells and they are stained with trypan blue. As soon as the cell dies, the trypan blue dye can go through the membrane and accumulates inside of the cell. So in a bright field image, the cell appears to be dark when it's dead, and it appears to be bright when the cell or when the membrane is still intact. So this is done thousands of times per day in a lab. So there are also specified systems that can do this for you. So for us, this just serves as a kind of benchmarking to see whether the system really works. And yes, it does. So we see clearly two groups, one group corresponding to the dead cells, one group corresponding to the healthy cells. However, and this is now the difference to a conventional, highly specialized instrument. For example, those group here, or this group in the middle, where you see these cells that are already kind of morphologically changing, which indicates that they are either dividing and doubling, or that they are going to die, they would be neglected in a conventional measurement. And with the techniques and the parameters that we developed, you can, for example, also be sensitive to these kind of cells, because there are applications around where you especially are interested in this kind of cells, and maybe you want to pick out those. As you see here, we can work in bright field and in phase contrast. Also dark field contrast is okay. And as we will see later, we will also be able to see fluorescent contrasts. So this is now from a paper that we published together with the group of Max Planck Institute for Medical Research. There we just had a look on what we can detect and what we can see. So as benchmarking system, we took a normal water in oil droplet generating chip, where we generated droplets of different sizes, different flow rates, and different interparticle or interdroplet distances. And we use the conventional high-speed camera with offline evaluation and the sensor with online evaluation in order to see whether all our parameters are correct. And as you can see here, they are all in one line, which means that the result of the conventionally determined parameters and the newly technique developed or determined parameters are the same. And this is also already something like searching for the edge of detection. So what we can do here, or what we showed, is here's 1,700 droplets per second that can be reliably detected. We can approximately double this if all the other parameters are perfect. So if the spacing, for example, is perfect, and you can ensure that you not have too many overlaps between the images. So this is kind of what is the edge. And one thing that you, for example, will never be able to see with a conventional high-speed camera is shown here. So this is a timeline of the droplet generation frequency. And what you can see here very, very clearly is that this changes over time. So once you see these small fluctuations here, which are in the order of maybe 10 Hertz or something like this. So these are directly feedbacks from pressure oscillations, something like this. That destabilizes your droplet generation system. And then you see this large scale variance here, which comes, for example, from deformation of your microfluidic chip or for creeping of your system. And this can be used then to feed this back into your system, and, for example, control your pump very precisely in order to avoid this. If, for example, you are interested in very, very uniform droplets. So this could be one application of the system. So now we're going one step further. So we're not only investigating how the droplets are produced, we also want to see what is inside the droplets. So this is a very, very basic example. So we have droplets, some of them are empty, some of them contain a cell. And the cell is almost 1/3 of the size of the droplet, so you will get a very, very prominent signal. So this is just a time signal with the, in principle, the squared contrast of your image. And as you can see here, so the cells are flowing by. And each time where you see a droplet coming through, you see one peak here. And then for the higher peaks, there's additional grayscale inside of your droplet, like here. And there you can be pretty sure that there's a cell inside. So using this very, very basic example, we verified the system and sorted according to this parameter. So at the first thing we did is we have here in green, this is our region of interest. In this region of interest, the droplet is measured and it is determined whether a cell is inside, yes or no. And in the same moment that this determination is done, we send a trigger signal to these two electrodes, and then they generate a high voltage, high frequency alternating field, and are attracting those droplets containing one cell. And when there's a droplet passing that contains no cell, they stay turned off. So with this, we can enhance in the outlet channel, which is the upper one here, the amount of droplets containing a cell to 100%. And we only have here 2% of false negatives. So only 2% of the droplets that contain a cell are wrongly not deflected. And we enhance this from a starting population of approximately 20%. One step further is we did this with a similar setup, but now we do not sort according to the content of the droplet, but we sort according to the size of the droplet. And here we wanted to enrich the small droplets in the upper channel and the larger droplets in the lower channel. And as you see here, this can be also done very, very precisely. The sorting rate in these experiments was approximately 130 droplets per second. However, this is limited by the sorting device. So if you can get for example, electrodes that can faster charge and decharge, or you go to completely different sorting mechanism like acoustophoretic or acoustic sorting or pressure-driven sorting, then you can increase also this this sorting rate. For us, as we are doing the detection part, the detection frequency is of interest for us. So this is how it's done. So you mark the region of interest here, and then you got a scatter plot with different cell populations here, where you can then in the software just click inside and generate a region in which you want to sort, and then you got out the sorting of your droplets. In a real experiment, this could be looked like following. So first, you can use Odin to directly control the production of your droplets. For example, you encapsulate cells, a specific number of cells, for example, inside of your droplet. Then you put it into incubation, for example, and then after several hours, you use the device, or you use a different device. So for example, a sorting, switching device here, in order to enrich some subpopulation of these droplets in one channel and in one outlet. And then you can go, for example, back to this and again to do this cycle periodically, and more increase or more and more enhance several things that you want to have. So I hope that I gave you now a short overview over what the instrument or what the technique that we are using is capable of. So now I want to show you some more measurements where we tested on how far we can go with this technique. As you can see here, so here we encapsulated large and small polystyrene particles inside of a droplet. And this is then how it looks like. So the droplets are flying by very, very fast. And the system has to classify how many droplets of, how many particles of which population are inside of the droplet. And you can see here that these, of course, it's an artificial sample. These are polystyrene beads, so they have a very good contrast. However, they do really good clustering. So you have here the empty droplets, then the single and the double occupied droplets occupied by the small polystyrene beads. And here you have those that are single occupied by the large droplets and doubled occupied by the large ones. And here, here, and here, you have those where large ones and small ones are both present in one droplet. And now, for example, the interesting thing is to sort out all those droplets containing one large and one small droplet, because you want to make some chemistry between these two. So this is a very important thing, because if you encapsulate such droplets, you are always limited by Poisson's ratio. So you will always get a mixture of all these possibilities. And if you only want one, you will not get by to a decent sorting device. So as I told you before, we can also go in the fluorescent regime, because sometimes it's necessary to have fluorescent information of your sample. So this is also done or also shown here. So we just took fluorescent particles. Of course, they are totally bright, and it's quite easy to distinguish between 1, 2, and 3 particles of the droplet. So this is no rocket science till now. However, if you're going one step further and having a look at real samples, this gets more and more tricky. So what we see here are way larger droplets. So they are now approximately 100 micrometer in diameter droplets. And you have cells inside that are approximately 8 to 10 micrometers. So the ratio of particle size inside of the droplet and the droplet itself gets very, very large. So, and the larger this ratio, the harder the detection is. However, for this ratio, we managed to find new evidence-based and empirical parameters that are very good in characterizing the amount of cells inside of the droplet. So you can see here, these are the two populations. So we labeled them by hand with the color. and you see how they do decouple if we apply these parameters. And now, for example, as I told you before, you can, for example, sort out all those blue ones that you can see here, or all the green ones, which are only once occupied. So the crux in this case is that all these cells look very different. So if you can see, if you look at the right-hand side here, so there are cells that are looking very, very bright. Some are more dark, some are round, some are already dying. So it's also very, very demanding in classifying them with your brain. So which is probably for image classification still the most or the best instrument to do this. So this is why we are really, really hardly working on these issues to get these detections right. Going one step further, we also did this in fluorescence. At the first sight, you would say, okay, of course, fluorescent, this is, you have more contrast, so this might be even easier than having it in a bright field. However, if you have a look at the right-hand side here and you really see the image, the fluorescent image, often you cannot say, so there are small dots, are these cells, are these dead cells, are these fragments? So the classification gets harder and harder the more demanding your sample gets. And this is in principle, how far we got till now. So we can already very, very precisely characterize, for example, the number of cells. Of course, this is not only limited to the number of cells, but you could, for example, also label just a specific subtype of cell, and you get then the answer of how many of these subtypes are included in your droplet. So, and I wanted maybe just as a statement is that here, of course, all these measurements here could be also done by a classical fax machine. So you could use classical fluorescent activated cell sorting. However, if you're already, so just to distinguish, for example, occupied and non-occupied. However, if you're already interested in how many cells there are inside, then those machines are already very limited. So this is a clear indicator for going to another system than the classical ones that are broadly available. So since I'm running out of time, I just want to go really, really briefly through this. So this is something totally different. I don't know who of you is familiar with microbiology. So as a sum up, you have a cell, you insert small particles inside of the cell, normally 1 micrometer polystyrene beads, and then you use a very high-speed camera and track the Brownian motion of these particles. And according to this Brownian motion, you can calculate back to the mechanical properties of the cell. And then there's this problem of either you can record at very, very high frequencies, so like 5,000 frames per second to get the short time properties, or you can record with 1 second interval in order to get the long-term answer. And with our or this is mainly the PhD thesis of Jonas. With his work, in principle, he was able to configure the optical sensor in a way that is possible to continuously record over 1 hour with 5,000 frames per second and continuously get the results out. So without the need of a huge hard drive or whatever. So you will just get the answer of the mean square displacement, it's called here, so of the mechanical properties of the cell for timestamps less than 1,000th of a second up to 1 hour. And yeah, so this was, for example, done by having a look at the tension in cell filaments when they are restricted to different forms. So there you can see, for example, if the tension is very, very strong because the cell is forced on a rectangular space here, this corresponds to a hardening of the fibers, which is seen in the lowering of the mean square displacement. So this is just another application of the system, but which is still very interesting and very promising. And at the end, I just want to show you something that we do in our lab if we are out of work, which is not very often. However, as it turned out, we can also use this Velomeer system that it's called to track a little toy drone with which we fly through our seminar room. And the system is tracking the small blinking light of the drone very, very precisely, as you can see in the upper right. And you can follow the path and the way of flight of the drone through the whole image with now 5,000 frames per second if you would be interested in. So with this small video, I want to finish my talk and thank you for your attention. And now I'm open for all kinds of questions.
Thank you very much. That was really awesome. There is a lot of questions in chat. Can you see chat?
Yeah, let's just open it.
If you could read the question before you answer, that's for the video recording.
Okay, so I start with Evan. So the question is, do you have any sort of environmental control, temperature and or CO2, or does it fit with regular microscope stage environmental chambers? Yes, we have, or we just, so in our lab, we also, of course, for the fluorescent imaging, you use a lot of lasers, so you have to encapsulate everything. And we just use a standard off-the-shelf environmental chamber. I think it's from the company called Picon, which you can put on your microscope and which encapsulates everything. So you can put in a heating device and CO2 management system. So this is like in just a normal size microscope or Neckernuss microscope environmental chamber. So nothing special, but you can use it. I hope that this answers the question. Then next question, are you doing demos? Yes, we can do demos. So you can just contact me if you are interested in this. We can do it online or on site, but I think we discussed this elsewhere. So, but thank you for the question. Can you use, next question from Lauren, can you use liposomes in the system too, or for now only droplet? So so I'm not 100% sure what you mean by liposomes, because I'm not so in this biological area, but what I can tell you is we investigated till now, we have cells, of course, all kind of droplets, bacteria, fibers, GUVs, so giant unilamellar vesicles, which I think is close to liposomes, I guess. Yeah, so something like this. So in principle, to sum it up, everything that you can see by eye with any kind of contrast. So either you can do it with a phase contrast or whether you can make it visible in fluorescent contrast, then in principle you can characterize it. So of course you're always limited to optical resolution. So the smaller it gets, the less information you will get. Okay, and I think this will also answer the next question. What's the minimum cell size that you can analyze? So we analyzed, for example, already E. coli bacteria, which have approximately 1 micrometer times 3 micrometers. So this in a non-fluorescent way. So this can be done, but this is also approximately the lower limit of the optical resolution. Of course, if you're just interested in whether something is bright or not, then in principle you can also see single quantum dots because they are bright enough. However, you will get no information about it other than yes, it is there. Okay, next question is what is the maximum cell size? So in principle, we are not limited. So as you saw before, we can also see a macroscopic object like drones. So this is just dependent on what kind of resolution you are using for your objective. So by using, for example, so the last images I showed you with this large droplets, which with 100 micrometer diameter, we used a 10x objective. However, if you go more, you can also investigate things like 200 micrometer or even larger. This is just a matter of optics that you use. And yes, GLVs we also already investigated. Okay. Next is from Pat Wesley. Is it possible to measure scattering to calculate particle size, or is particle size only based on imaging? From our technique, we do not use conventional scattering techniques, so we only use image-based techniques. So there we are limited to what we see and what we can measure. So we have no scattering, might it be fluorescent or just with, I don't know, conventional scattering methods we have not included. And yeah, I think it's just a different kind of technique. So could this be used in stop flow mode for imaging dynamic processes like cell division? So in principle, yes. So of course you can go down with frame rate. However, I mean, this is not what the technique was meant to be. So to be honest, I guess that there are way better systems to use in stop flow mode and imaging processes that are on a very large time scale, so like the cell division. However, for example, the last thing that I showed you with the tracking thing, of course, there you can detect the, for example, you can track the nucleus of the cell if it's dividing, for example. This can be done. So I don't know which kind of application you have in mind or which you think might benefit from this, but yes, in principle it's possible. However, it's probably not the best technique to use for. Okay, yes, Juvis. So the next question is from Karin. Do you think there's capacity to automate the whole process? Image and analyze and implement some machine learning to inform information of next batch of sample. Is there always human input necessary? No, we also have implemented something that some people might call machine learning. I think it's, so what we are doing is we can do, for example, principal component analysis in order to get out the most significant parameters. If you're doing, for example, if you're classical learning approach, so you divide by eye the group into parameters or into two subgroups and label them in good and bad, for example, then the program is able to tell you which parameters you should choose in order to get the best separation. But of course, implementing machine learning and deep learning and neural network will be the next step in the evolution of the system. This is planned, but completely AI-based sorting is not yet implemented. So yeah, I hope that this is kind of answering the question. So Kate, you just interrupt me when we should finish with the questions, because there are still some clinging in. So I just continue till somebody stops me.
I think please keep going.
Can you sort as well, or is this just imaging? So so the system that I showed you is not sorting. The system is analyzing and triggering a sorting device of choice. So we can, in principle, work together with each and every sorting device. So this is the good thing, because from our experience, there are groups outside who have really outstanding capabilities and are really good in developing new sorting devices and improving standard ones using 2, 3, 4, 5 electrodes or other techniques. So this is very, very special. And there are also sorting things that are more appropriate for some samples and others are appropriate for other samples. So that's why we just focus on telling the sorting device when it should trigger an event and when it should start sorting. So I hope that this is kind of okay for you. So I see this was the last question.
Thank you so much. I would be really excited to give it a try. I'm especially happy to see that you can do vesicles, because I think a lot of people in our community would be interested in something that works with unilamellar liposomes of approximately cell size. So that's really cool.
So we are constantly, or we are currently working together with our partners from Max Planck Institute. I think they're also, I think through these guys, the contact was a little bit established through those. We slipped into this synthetic cell thing, and together with them, we are working on something, regarding GUVs or SUVs or whatever in this region.
Thank you so much. Thank you again. Thank you very much for the seminar, and thanks everyone else.
Yeah, so thank you for having me and for everybody else. So have a nice day. So stay healthy and safe, and yeah, have a nice day.
Same for you. Have a nice evening. Bye, everyone.
So, thank you. Goodbye.